Hi, my name is Satnam Saray. I'm a third year PhD student and I'm working here at the Conway Institute of Biomolecular and Biomedical Research in University College Dublin. My project uses computer simulations and biochemical methods to engineer new therapeutics or medicines to protect against diabetic nephropathy. Diabetic nephropathy, also known as diabetic kidney disease, is a complication of diabetes and is the leading cause of end-stage renal disease or kidney failure worldwide. Approximately 25 to 30% of patients with either type 1 or type 2 diabetes will get a form of kidney complication. With the increasing rates of diabetes and obesity amongst an ageing population, diabetic nephropathy is set to increase its burden on healthcare providers across the globe. Diabetic nephropathy causes progressive damage to the kidneys. The kidney is the organ responsible for filtering out toxins from the blood. It is made up of hundreds of thousands of small filtration units called nephrons. At the tissue level, this is reflected by the progressive degradation of the nephrons and the increased size and thickness of the extracellular matrix surrounding the nephrons. There are many mechanisms that contribute to the progression of diabetic nephropathy linked to diabetes. Risk factors for getting kidney complications with diabetes include high blood pressure, an imbalanced diet, such as too much sugar, age, body fat percentage and ethnicity. Current treatments for diabetic nephropathy include angiotensin converting enzymes, or ACE inhibitors, and angiotensin II receptor blockers, also known as ARBs, which reduce blood pressure. However, these do not reverse the effects or protect against the damage caused by the disease. Here, in the Diabetes Complications Research Centre, it was discovered that the level of gene expression for a protein called gremlin was very strongly correlated with the progression of diabetic nephropathy, as there is little or no expression in healthy human kidneys and maximal expression at end-stage renal disease. Hence, we think if we can somehow mop up the extra gremlin produced, we could potentially protect against the progression of the disease. The way I'm trying to do this is by performing computer simulations on the structure of proteins that have been found to interact with gremlin and design new proteins that strongly bind and nullify its effects. Protein structures are experimentally derived and contain information about the location of each atom that makes up the protein to give a three-dimensional blueprint of the protein. Proteins are made up of strings of amino acids that fold up into beautiful and complex structures that can then perform various tasks in the body. I've built a program that takes a protein structure file and calculates the stability of the protein using quantum mechanics and estimations of chemical interactions. It's basically a lot of maths. This gives us an energy score and can tell us how comfortable a protein is. If a protein is unstable, it will have a high energy score and won't fold properly. But if it is stable, it will have a low energy score and probably will fold properly. Hence, we can take an existing protein structure and change amino acids at important positions to change the properties of that protein. So in this case, we have taken the structure of bone morphogenetic protein 7, also known as BMP7, which is known to bind gremlin weakly, and have introduced mutations that the program has predicted will increase binding strength to gremlin. The cool thing about this is that we have also designed BMP structures that not only potentially bind gremlin more strongly, but those same mutations decrease binding strength to other proteins, hence aiding specificity. The next stage of my PhD is to test these computational predictions in the lab to see if they actually work. Owen is a postdoctoral researcher in the Diabetes Complications Research Centre who will be helping me carry out these experiments.
I think Sat's work is very important as far as our research. We can ask questions based on our experimental data, test it out by in silico analysis and computational modelling. The advantage of this to us is that it's obviously we can directly address the hypothesis we have. It's certainly a lot quicker, more cost effective, and hopefully in the end we can address the sort of questions we have with regards to our disease of interest.
Using these structure-based energy calculations in this way is a new and exciting way to carry out research. These computer simulations help us to decide which experiments to actually do in the lab, and it's a great way of decreasing the time of therapeutic development and decrease the cost, but it also helps us to see how good our models actually are. The more we do this, the more we hope we can improve the computational models. Thanks to the Innovation Academy, and also thanks to the Irish Research Council for my funding.